So today we're going to talk about chapter 14, and basically it is epidemiology, or some of the sort of ways that we analyze and track disease. I also like to call it the chapter of many definitions because there are like 300 terms that you need to learn. So before we do all of that, we should talk about why we care about this particular field and this particular approach to studying disease. So it helps us to understand what specific agent causes disease. Once you know that, that will help you treat it. It also tells you how the agent that causes the disease is actually working in your body. And again, that's very useful for therapeutics if you know which cells to target. And it talks about how agents are spread, which can be helpful in a variety of ways, including public health campaigns to reduce disease. So I just want to remind you of something that we talked about in the very first lecture, I think, or maybe it was the second one. It was the first one. Koch, this German scientist, and basically he was really instrumental in sort of laying out the way we can connect a causative agent to a disease. And he developed these principles when it was just sort of beginning to be understood that microbes can cause disease. So here are his postulates. And what I want to say before we walk through them is that they're very useful. These are his original postulates, but they don't allow us to answer the question of sort of what is going on with every single disease. And we'll talk about the situations where this is not completely helpful. So his postulate first, in order to show that this agent causes that disease, was that the same pathogen, that is the same causative agent, must be present in every case of the disease. So let's consider this. Autism. We don't yet understand what causes autism. Now, if we were going to say it's a microbe, what we would then have to show is that every autistic individual is infected with the same microbe. Now, clearly that's not the case, or at least if that is the case, we haven't identified the microbe. But if we wanted to say that the same causative agent is causing autism, then there has to be that same, whatever it is, exposure to pesticides, vaccines, which now have been debunked, but whatever it is has to be the same in every case. 2, the pathogen has to be isolated from the diseased host and grown in pure culture. Okay, so you have the sick person or animal or whatever, you isolate their blood or tissue or bodily fluids, wherever the pathogen happens to be, and then you grow it in pure culture to prove that it's actually what it is. Then you take that pure culture that you've generated artificially in the lab, and you inoculate it into a lab animal, into a healthy, susceptible lab animal, and you see the disease replicated in that animal. Then you isolate tissue or fluids or blood from that animal and show that it also has. So these are, this is sort of a lengthy process. Here's an example of that working, okay? So if we're studying, for instance, pneumonia, and we get a pneumonia sample from a pneumonia patient, then, or from our, for instance, our sick mouse. We have our dead mouse, our diseased mouse. We isolate from their bloodstream or from whatever tissue or bodily fluid we're sampling. We grow it in pure culture. We look at it under the microscope using, smears and staining. We inject it into a healthy animal once we've cultured it. And then we see that this new healthy animal has the exact same clinical symptoms. You isolate from that animal, again, back into pure culture and look at it again, and it's the identical organism. Now, there are some problems with using this approach. So, for instance, some pathogens cause different diseases in different contexts. So that's an exception to this rule. Some of them also cause disease only in humans. So you can't do that side animal model thing, and it's not really ethical to inoculate people on purpose unless you're doing, you can do it for things like the cold virus, but that's about it. And then also, if you're requiring that you be able to culture it so that then you can take that culture and inoculate a sample, you have to be able to culture it, and many pathogens are not easy to cultivate. Okay, so here is the start of some of the many terms. Pathology is the study of disease. Etiology is the study of the cause of that disease. Okay, so pathology is kind of a broader topic. Pathogenesis, which you know from your oral presentations, is the development of disease and really thinking about it from a cell biology perspective. Which cells in your body are infected, what happens inside those cells, what proteins are involved inside the cell, how does it spread from cell to cell, what are sort of like the progression of the disease. Infection is colonization by pathogens, and disease, definition of disease, is an abnormal state in which the body's not functioning normally. So it could be, you know, something that's not caused by a pathogen necessarily. Now, a couple of things, and we'll come back to this, and we've also talked about it already, but we need to sort of consider normal microbiota. And the difference between that and transient microbiota is that normal microbiota is something that permanently colonizes you. Basically, from the time that you first come into the world, most likely you were inoculated during childbirth. And these days, if you do a C-section, you inoculate the baby with microbes so that they actually they're colonized. But usually, as you're being born, you're being colonized by your mother's microbes. And that's your sort of normal microbiota. Transient microbiota can happen where you, for a few days, weeks, or even months, you have something that's in your body. This term symbiosis is the relationship between normal microbiota and the host. And there's a couple of examples of that we'll talk about in a second. This is just some pictures to show you. This is the cool factor. So these are things in your body that are normally supposed to be there that look kind of weird and frightening. So here's in your nose. So these round orange guys, these are all pseudo-colored electron micrographs. They're scanning electron micrographs, so you get that three-dimensional look. So we've got bacteria here in your nasal epithelia. So these little fingery things are normal projections from your cells inside your nose, and these are microbes growing in your nose. That's totally normal. In the lining of your stomach, we've got other microbes. And in your large intestine, in your colon, we have lots of different species of bacteria. So what is symbiosis? Many of the normal microbes in our bodies are in symbiosis with us. There's several types, and you just have to memorize these. You need to know these terms. That's part of taking this course. So I'm just going to tell you what the definitions are, and you might see this on a test matching question, possibly. So commensalism is one where one organism benefits and the other is unaffected. So meaning there's no positive or negative for that organism, it doesn't get harmed. Mutualism is where both organisms benefit, and both of these are examples of symbiosis, just different kinds. And then parasitism is where one organism benefits at the expense of the other. So one gets a benefit, the other doesn't. And we have parasites that, for instance, if they live in us, they get nutrients and stuff like that, and we kind of get sick. Now, there's this other term that we have to understand, which is opportunistic pathogen. So some of the normal microbes in our bodies are opportunistic pathogens, which means that in the place where they're normally found, they're either harmless or beneficial. Okay? Can you think of an extremely common harmless/beneficial organism in your intestines? E. coli. Now, what happens if E. coli gets into your urinary tract? You have an infection. So clearly it matters where this organism is. In some parts of your body, or if you were on a surgical table and they're doing whatever, right? And some of your skin bacteria got into the surgical site, then that could cause an infection that would be very bad for you. So here's just where and what kinds of things are found. Normal microbiota are found in the eyes, mouth, upper nose and throat, on the skin, in the large intestine, and the urinary and reproductive systems. Basically your mucous membranes and the surface of your body. So there's also a few other terms to know. Microbial antagonism is competition between microbes. I think that's sort of self-explanatory. Now, what are microbes, the normal microbes, doing for you, right? Remember, they're taking up space that might otherwise be colonized by pathogens. So just by being there and not being a pathogen, just taking up the space and using nutrients, they're preventing, say, E. coli O157H7 or C. difficile from colonizing. They produce waste such as acids, which can actually be good because some parts of your body need to be acid. And they produce antibacterial chemicals. that they themselves are immune to, but they kill pathogens, basically. They're not doing it out of the goodness of their bacterial hearts. They're doing it because they're in competition. They want to keep their real estate, and they're like, this is mine. You can't come here. Probiotics you've probably heard of. This is live microbes that you take as a supplement in order to have a beneficial effect. So for instance, some people who have had their normal intestinal flora destroyed by a long antibiotics course or something might take probiotics to restore it. Okay, so let's get to some of these other terms. I told you it was the chapter of bazillion definitions, and I'm not kidding. So a symptom is a change in body function felt by the patient. So let me ask you this. You go to the doctor and you have chills. Is that a symptom or is it a sign? Now, what is a sign? A sign is a change in the body that can be measured or observed. Okay, so you go to the doctor, you feel things, the doctor can see things. On occasion, those things are the same. I'll give you an example. If you have a fever, you feel feverish. And they can also measure that. They can take your temperature. But if you have chills, which one is it? Is it symptom or is it a sign? It's a symptom. You experience the chills, but there isn't a really easy way for a doctor to measure your chills, okay? Pain is another one. Pain is a symptom, which is very difficult to measure. You can ask somebody to describe their symptoms and record it, but you can't, like, hook them up to some machine. I mean, possibly with brain scanning technology, that may be soon possible, but it's hard now.
So you said fever was science.
It can be, yeah, because I would argue that usually fever in biology and in medicine is used clinically to mean you can actually measure that someone's temperature is altered. That would be a sign. But if you say, I feel feverish, that is a symptom. Does that make sense? Usually when we use it in this context, it's actually a sign. Because fever is a real biological thing that you can measure. All right, syndrome is a specific group of signs and symptoms that accompany a disease. So here are the stages of a disease. Basically, we start with the incubation period, okay? Now, what is graphed here is on the y-axis is the number of microbes. So if it's a virus or a bacteria, it's infecting us and then it's multiplying, right? So we can measure how many of the viral particles are in your blood or how many bacteria or whatever. So initially you get whatever dose, and initially as these guys multiply, there's an incubation period during which you have no signs and symptoms. Okay, so you wouldn't know that you are sick. You might suspect it because your daughter coughed in your mouth, right? But you don't know that. Even though you already are infected and you already have this increase in microbes. The next stage is the prodromal period, which means what? Some of you have probably heard this term. What does it mean? Prodromal. Have you ever heard of this in the context of a cold sore? So what does that mean in that context? Do you remember? So in the context of a cold sore, it would be like tingling or some sort of early symptoms that you feel and you're like, I think I'm going to have a cold sore in a couple of days. The prodromal period is still the number of pathogens is increasing. You're starting to get symptoms, but they're mild and they're different from the full-blown disease. Okay, so we pass from those two periods into the actual illness. You have the whole suite of symptoms. There might be a progression, but it's the most severe part. And during that time, the number of the pathogens is increasing. Then your body kicks in and you start to fight back, and then the number goes down because your white blood cells are consuming the bacteria and your antiviral chemicals are working. And then there's a decline in your signs and symptoms, followed by convalescence, where you're recovering from having just been through that horrible experience. So there's a couple of ways that we classify disease, and it's important to understand the distinction. So acute disease, and we've talked about this before, acute disease is when symptoms develop rapidly, and they usually then also have an equally rapid decline, right? So the whole thing is a short period of time. A chronic disease where the disease develops slowly and you have it for a long time. And then there's this term, a subacute, where the symptoms are somewhere in between. I just want you to know that I would expect you to be able to tell me about acute and chronic, or given an example, tell the difference, but I don't usually test on subacute because I feel that's a confusing term. You should know it, especially if you're going into medicine. Latent disease I would test you on. So this is where you could have, an initial acute illness, then it goes into, a period of no signs and symptoms for some extended period of time, and then you have recurrences. So there is some period of time between first getting sick and then whatever recurrences you have to later there's no symptoms. You could even have a latent illness where there's no symptoms, but you still have the pathogen in your system. Okay, we also classify diseases by the order. So primary infection is the acute infection that causes the initial illness. What do you think a secondary infection would be? Have you ever heard of that term? Let's suppose that you get a virus. How about you get some sort of cold? It's nasty. It lasts for 5 or 6 days. Then you get better. And then, 2 or 3 days later, you have the same symptoms again, only this time it's not a virus, now it's a bacterial infection. Because you had this initial cold that kind of reduced your immune system and things happened and bacteria got where it wasn't supposed to go, and now you have a sinus infection. That sinus infection is a secondary infection that happened in part because of the first one. So it's an opportunistic infection that happens after a primary predisposing infection. Can you think of a really good example of a primary versus a secondary that's famous, that's the result of you having a really compromised immune system? Yeah, exactly. So having HIV and then developing the acquired immune deficiency syndrome is the primary infection. Then you can get shingles and weird pneumonias and weird cancers and all kinds of other things as secondary diseases that are the result of having this reduced immune system. Okay, a couple more terms, types of infection. A local infection is where the pathogen is limited to a small area of the body. An example would be like a paper cut, when you get, you have some sort of skin infection and it gets red and it's annoying, but it's not like it travels to your heart. Systemic infection is where the microbe or toxin spread through the whole body, through the blood or the lymph. And so even in some cases, you can have, for instance, a local infection with the bacterial agent, but if it makes toxins, especially if it makes an exotoxin that while it's alive, it's making and it's getting into your bloodstream, even if the bacteria themselves don't travel through your body, the toxin can go through your whole body and make you sick, or the microbe itself could go through your whole body and multiply in your bloodstream. So either the microbe or the toxin, if it's spreading through your whole body and causing symptoms everywhere, then it's considered systemic. Focal infection is one that begins as a local infection and spreads via the blood lymph. So it goes from local to systemic. All right, so now we need to talk about how the disease, different ways we sort of classify diseases depending on what happens in the body. So sepsis is when we have a toxic inflammation arising from the spread of microbes, especially bacteria, or their toxins from a focal infection. So again, that spread through the whole body is considered sepsis. And it can be either that you're full of microbes, like your bloodstream is actively full of bacteria, or it's full of toxins. Septicemia is specific. It is specifically a systemic infection that's coming from the bacteria or the pathogens multiplying in the bloodstream. So we can have bacteremia, that's bacteria in the blood, toxemia, toxins in the blood, and viremia, viruses in the blood. Okay, now spread of disease in the population. So communicable disease, that's a disease that's spread from one host to another. Are there diseases you can think of that would not be communicable diseases but are nonetheless caused by microbes? What about botulism? When you get botulism, can you spread it to anybody? No, you could spread it back to the earth, where it came from, and the earth does not get sick. Or tetanus. Okay, so a contagious disease is one that's easily spread. So this is sort of a little bit of a judgment call, but you know, a cold would be contagious, I would say. AIDS is contagious as long as you're, you know, sexually active or sharing needles with infected drug users or, you know, getting blood from wherever. It is contagious. But something that's very hard to spread wouldn't be considered contagious. Non-communicable disease. A disease is not transmitted from one host to another, so we already have talked about some examples of that. Okay, so what makes you susceptible to disease? What do you think? Yeah, reduced immune system. It's compromised for whatever reason. Anything else? Say again? Yeah, absolutely. Yeah, just being in poor health overall. Yeah, being elderly or sometimes, depending on the disease, right? So some diseases are more common in elderly or children, some are not. There's a few you may not have thought of. So your physiology can play a role because the urethra in women is short compared to the men. We get more UTIs. Okay, so that's a predisposing factor. Genetics, so some of our genes can make us more readily sick or cause the disease directly. Current physical conditions, so illness or things like chemotherapeutics, which reduce your immune function, and demographics, age, and the things you do, the choices you make, smoking, and that kind of stuff. The factor of weather and climate is actually really complex, but one simple way to think about one of the parts that this plays is that it influences what microbes are actually present or what vectors might be present, right? Because, for instance, if we're talking about one disease that's transmitted by ticks, then you're going to be more susceptible if you're living where there's deer and, you know, shrubbery and stuff like that. Now, I'd like to sort of take a bigger perspective because all of those are great examples, but they really kind of leave out some of the important factors that in this part of the world we don't really think about too much. So here are the leading causes of death by infectious disease in 2002. And what I'd like to point out, which we know from having read our disease list A, is that most people in the world are dying of lower respiratory infections. Imagine that, like every time you got like some sort of cold-like thing, you were like, oh, I might die today. AIDS is killing a lot of people worldwide. Diarrhea, tuberculosis, malaria, and measles. So all of these are things that we don't actually really worry about too much. Why is this happening globally, and what is different sort of in the Western world? So one thing is water and sanitation. So diarrhea is actually deadly if it's untreated and you already don't have access to, you know, a lot of fluids to rehydrate you, and if your exposure dose is really high. So there's, say, for instance, 2 million children worldwide that die from diarrhea that could be prevented just because they don't have clean water. And then there's a whole host of things that don't kill you, but they'll cause pretty significant impacts on health because of this lack of water and sanitation. Pollution, so different parts of the world, if you're in an urban area, you might have higher pollution, but you know, what people don't really think about is if you're, for instance, cooking over open fires, which is in more traditional cultures sometimes the case, that actually increases your risks. And also you'll notice that in those regions, guess who's most impacted? The people doing the cooking. Also poor nutrition. So again, this is something we probably don't think about too much just living here, but worldwide that this is quite a serious problem. So again, and it particularly impacts children. And education, right? So what your sort of social conditions are can really impact your health overall. The other feature is access to resources. So great many of the countries in the world have a very reduced access to resources. Half the world's population lives on less than $2 a day. That's pretty sobering. Access to resources is, you know, being able to buy medical care and being able to buy food. And the other thing that a lot of people forget is good governance. So if you can't trust your government to manage your resources, or you're living in a really unstable area where there's a lot of you're constantly moving because of instability, wars, in unequal distribution of resources, then that will affect your health significantly. Now, in the U.S. Is an interesting sort of example of how all these factors sort of come in, because if you look at the number of deaths in the U.S., from the early part of the 1920s to present day, there's been a dramatic reduction in mortality due to disease that is directly correlated to changes in all of these factors. So what are some of the things that were done? I don't know if you have read any of the sort of prairie books that are sort of classics in American culture, but Little House on the Prairie, for instance. I don't know if you've read that or aware of that, but this is a book written by somebody who was in the U.S. Midwest, a couple hundred years ago, and at that time malaria was really common, which we now think of as an African disease, but it's something that can happen wherever there's mosquitoes, right? So what happened in the 1920s was that, for instance, in Virginia, they started draining a lot of the standing water, which reduced the mosquito populations. They also started spraying insecticide from aircrafts. And consequently, the mosquito population went way down, and malaria transmission also went way down. I'm not saying that that didn't have any other consequences for the environment or for people's health for other reasons, but this is just one thing that did make a difference in terms of that specific disease. The other thing is that at a certain point in the history of this country, people started filtering water, and then they started chlorinating it. So wastewater treatment and just drinking water treatment really dramatically changed, especially the transmission of salmonella, which is what causes typhoid fever. It used to be really common. My grandmother's oldest brother died of typhoid fever. She nearly died of typhoid fever only just 100 years ago. And that is no longer something that we even think about, but it is caused by salmonella from untreated water. And there are also medicines that have played a role. So, for instance, measles cases have gone down as a result of medications, and that specifically because of vaccination, right? So measles, pertussis. But what I'd like to point out is that a lot of times the conversation superficially in healthcare is about how important vaccination is, right? To keep at bay things like measles. And it is important, but to put it into context, so this is a graph that shows you over time, right? So to the 1960s, which was really when these measles and pertussis and so whooping cough and stuff like that were starting to get under control finally, when they had been really high in the population. So the 100 years preceding that. And on the y-axis, the number of people dying. And this is just in Britain. So, they identified the cause of aging around the turn of the century. And this, in the 1950s, is when widespread immunization happened, which did reduce mortality a little bit, right? And that's not insignificant if you want to keep all of your children. But this is the result of changes in sanitation and changes in understanding of disease and different kinds of economic factors and things like that. So it's pretty important to understand that, because this is the difference between the first world and the developing world. All of those simple things are really what's killing people globally. It's not that they don't have a vaccine.